Okay, so hi Ben, how are you?
I'm good, thank you. How are you?
Yeah, good, thank you. Not too bad. What have you been up to so far today?
Just focusing on writing up my my master's dissertation. So yes, this cuppa is a welcome break from it.
Good. I'm glad it can offer some a bit of time to relax. That's exciting though. The end is in sight.
It is indeed, yeah.
Cool. So do you just want to give a bit of background about which specialism you're on? Guessing you're in third year, but yeah, just kind of a bit about that.
Yeah, okay. So I am a trainee clinical scientist in Public Health Wales. So I'm on the Clinical Microbiology Scientist Training Programme. Microbiology is a discipline which looks at bacteria, viruses, fungi, parasites. But as part of the training, we also look at things like health protection, epidemiology. We've got a food, water, and environment discipline. And then some specialisms, things like antimicrobial chemotherapy, anaerobes, and pathogen genomics as well. So it's a fairly broad discipline.
Yeah.
For microbiology, but so yeah, as you said, I'm in my third year now, focusing on writing up my dissertation. Hopefully, looking to finish towards the end of the summer, September time. Yeah.
Yeah, that sounds like I obviously knew that it would kind of be bacteria and viruses and fungi and stuff like that, but it actually covers a lot more than I would have guessed.
Yeah.
You must see quite a lot of different referrals coming through of patients and things like that. What are kind of the main things that you might see?
Oh gosh, it COVID. That's it. COVID is all I've seen now for months and months and months.
Oh.
But I mean, there are lots of different things in all the different disciplines. So, in bacteriology, it could be anything from things like urine cultures to blood cultures, sputum samples. They also they get tissue samples. So you could get bits of bone. Pathology will get lots of nail and hair samples looking for fungal infections. You'll sometimes get sort of things like, I don't know, an amputated toe just in a pot to have a look at, and then go into virology. Virology, lots of swabs. Obviously, that's what we've been doing for COVID. So when you're doing molecular testing, lots and lots of swabs, but also things like blood for HIV testing, hepatitis testing, cerebrospinal fluid. If you're looking at sort of meningitis infections, that sort of thing. And they also get other samples like urine, and then I think any disciplines that I've missed, and then you've got the sort of parallel side then, which is the health protection side, which is outside of the laboratory, and it's working with epidemiologists, things like contact tracing, which people hadn't really heard of prior to COVID, and now everybody knows everything about. And the bioinformatics side, that's a real challenge. That's a discipline in itself, isn't it? So yeah, that's all really broad. And then completely different. Then we've got the food, water, and environment laboratory, where you could go from testing a cerebrospinal fluid looking for enterovirus on the Monday, and then on the Tuesday you are testing a sample of raw milk for Listeria. So it's it really is really broad, which is good. It's good. So it certainly makes it interesting. Yeah.
Never doing the same thing twice.
Yeah, you're definitely not doing the same thing twice. It does make it hard because when you complete the portfolio, you need to see it, and then you need to get your DOPS done and get it signed off so that you can leave behind the cerebrospinal fluid and then start testing a sandwich or sausage roll. and then you're back in the other lab, and then there's a toe there, or
That's insane.
Yeah.
Do you kind of rotate around all of the different areas then, or within your specialism, is it just kind of the hospital bits that you do?
Yes, you do go around all of the different disciplines. In your first year, there's a focus for us on bacteriology, virology, health protection, and epidemiology. And then the fourth one is just all of the different specialist reference labs. So within Public Health Wales, we've got things like the UK Anaerobe Reference Unit, which is a reference unit for anaerobic bacteria like C. diff. And then there's the Specialist Antimicrobial Chemotherapy Unit, which is a lab which looks at resistance, resistance of the organisms against antibiotics, but it's a much more specialist lab. We've got a Cryptosporidium reference unit, a Toxoplasma reference unit. So you just go around, and that first year really is about getting a feel for the entire service. And we're quite fortunate because within Public Health Wales, we are, unlike Public Health England, Public Health Wales is an NHS trust, so we're not civil servants, which is PHER. So it means that we're in hospitals, we're integrated within that service, but also we're not tied to the local health board. So so for example, I'm based between the University Hospital of Wales and University Hospital Landoc, which is covered by Cardiff and Vale Health Board. That's the local health board. Now, if I was working somewhere like Blood Sciences, then I'd be employed by that local health board. But Public Health Wales sort of occupy parts of the hospital right across Wales, which is really important when you're looking at things like infection, to be able to look at trends right across the country.
Yeah.
And it means that, I've had experience working in Swansea, because that's where some of the reference laboratories are. But also hoping to get up to North Wales as well, and to see how that service looks. Obviously, I think it's a smaller service, a smaller number of staff. So as a result, they need to have a wider skill set, rather all the sort of the specialists and reference labs that we've got in Cardiff.
So it's more of like a whole country view then. Within like genomics, we have genomics laboratory hubs, so like a main and then the satellite spokes. So it sounds like you're kind of more holistic into the view of the whole country.
Yeah, absolutely. It is. It's it is one sort of Wales-wide service. That provides everything, and it's really good because we've got that sort of health protection element as well. So it's a standalone laboratory role, where you're just sort of dishing out results to individual patients, which is still extremely important. But you're also having that opportunity to take a step back and see what's going on right across the country, which obviously during COVID has been critical.
Yeah, definitely.
So yeah, that's one really big advantage that we've got.
That's good. So what's been your, seeing as you've covered so many different things, what's been your favourite so far?
Oh, this is going to be a challenge, because if any of these laboratory managers see this video, I'm actually going to go with the Food, Water and Environment Lab. That's where I've chosen to do my project. It was up to us where we chose to do our dissertation. So prior to coming on to the STP, I'd done my bachelor's and my master's in biomedical science, and I'd done a PhD in biomedical science, medical microbiology. So it was I had a relatively strong research background coming into the STP. Something that I really wanted to get out of this project and was important to me was that I'd done this sort of blue sky research, where you can just sort of ask an academic question and sort of dig around and see if you can find an answer. And it was really important that I had the opportunity to do a project which was sort of service delivery focused and had an impact on the community, or on patients or service users. So Food and Water offered that. So I came from a sort of a molecular microbiology background. I'd had the opportunity to learn lots during my rotation. So when we're looking for things like enterovirus, herpes simplex virus, varicella zoster virus, cytomegalovirus, there's lots and lots of viruses that we look for by PCR, by molecular. And the same in bacteriology, they're starting to move over to panel testing, multiplex panel testing. When I went to the food and water lab, everything was still culture. So still growing everything on agar plates. And they, much of bacteriology, both clinical and health protection in food and water, is still culture. And that still plays a really, really important role. But when we need a rapid turnaround, there is an opportunity for molecular to step in and give you that rapid turnaround. Culture still remains that gold standard, but in some situations, that rapid result really can make an impact. So I approached the scientific lead, Debbie Charles, who was extremely supportive, and we were looking at Listeria monocytogenes as a model pathogen for what we could do. So we were going to look to see if we could get a PCR set up for Listeria monocytogenes, and use that as a model really going forward to say, right, well, will we look to introduce testing for other food pathogens in future? And it was really useful because I was able to bring that experience from a clinical setting and take it to this health protection laboratory, the Food, Water and Environment Microbiology Lab, and apply it there. And as a result of the project, we do now have sort of a validated workflow for being able to for the detection of Listeria from food samples. So Listeria's got quite a high fatality rate in vulnerable categories, so immunocompromised people, and it's also got quite a high rate or relatively high rate of causing termination in pregnancy, so leading to an abortion, which is why it's of public health concern. It's much rarer than some of the other food pathogens, but when there is an outbreak, it is really important to be able to get that result quickly so that you can contain it. So having that option was really important. So there's still a little bit of work to do to make sure it's fully ready to be up and running. But we've got a sort of proven workflow now that's able to be implemented and is a model really for going forward for other food pathogens. Not for routine use necessarily. So, we're still going to be using culture for most samples, still growing them. But it does reduce the turnaround time by a significant number of days, which means that, if you have got an outbreak or something like this, then you're able to get that rapid result. So that's beautiful. Yeah, and another experience I've had within food and water was we were looking at endoscopy rinse water. So, coming from a clinical background, I'd done biomedical science. I wanted to be a clinical scientist. I had absolutely no interest in food science. If I had, I just studied food science. But actually, I've learned to sort of appreciate that sort of role it plays in health protection. So they also test waters there. One of the waters they test is endoscopy rinse water. So used in things like bladder cancer, you've got an flexible endoscope, which obviously passes up and can have a look around in the bladder. And I'll leave that to discipline to be able to explain that one. But they'll then put it into a special cleaning machine. And then this machine, I'm probably going to undersell what this machine does, but in layman's terms, it passes through sterile water, and then that sterile water is collected then. So after the cleaning cycle, sterile water goes through the tube, we collect it, and then we'll test it. And that water should still be sterile coming out the other end of the tube. Now, sometimes it's not. And if it's not, that cystoscope needs to be taken out to use. It's not safe to be used on a patient, so it'll be rerun. And they were having these samples where they were finding that there was unsatisfactory microbial sort of contamination. And of course, it was taken out to use, so there was never any risk to patients in terms of that. But we were looking at whether or not we needed to to look to solve the problem. Where's this sort of contamination coming from? And meeting them with the estates team, meeting with the nurses that were, performing this sort of this operation, this technique, and understanding what the impact was. Sort of, it really brought out to me what the role of that laboratory is. So yeah, that was really interesting, and sort of investigating the source. We found the source, and we were able to put that instrument back into use, without causing any risk to any patients, but also making sure that they were able to continue providing that service. So I think, yeah, I would probably say the food and water has been one of the most, surprisingly for me, has been one of the most interesting parts of my rotations.
That's really good. I feel like it's always the one you don't expect.
Yeah. Yeah, I certainly didn't expect it. I'll be honest with you, and I've said to Debbie that, when I was looking at my rotations in the first year, you're thinking, oh, I'm going to the specialist antimicrobial chemotherapy unit, and I'm going to the UK anaerobe reference unit or the Welsh Specialist Virology Centre. I wasn't particularly excited when I saw that I was going to the Food, Water and Environment Lab. That wasn't for me where a clinical science is at. But I was very wrong. I was very, I've learned so much from being down there. Yeah, really, really interesting. So yeah, definitely caught me out with that one.
Wow. So coming from like a genetics background, I know that with like sequencing and stuff, if you took a water sample, you're limited with what can then grow in the culture lab. You're not going to sequence everything, you're not going to find everything, because only certain ones will grow. So is that something that you have to take into account, or is it just like the main ones are fine for culturing?
It depends what you're looking for. So in a lot of the waters, they do Legionella testing. So that's specifically what they're looking for. They don't do any sequencing on their waters.
Yeah.
But that was what you asked, wasn't it? Sorry.
Yeah.
Was what? Yeah. So lots of it's Legionella testing, and one of the big problems you've got with it is that you need to test a fairly large volume to be able to detect it. Now, working in molecular, where you sort of, pipetting 10 microliters, and then, go into food and water where you could have 10 liters to filter. So they filter it through onto this little pad. So you've sort of almost pulled out any bacteria out of this 10 liters of water, or, however much they're testing, onto this little disc. And then that disc does go onto the agar, and then will grow. The Legionella turnaround time, though, is around 15 days. So that is quite prolonged. We are looking at whether or not you could have a PCR for Legionella. I mean, there are PCR systems on the market, but just whether or not they fit for use for what we need, and whether or not they're sort of economically viable and give you that benefit that you need out of it as well. Yeah, I mean, most of the sequencing is done in the clinical lab. So that's the pathogen genomics unit that we've got. And I will probably avoid talking too much about that with you, because you are a geneticist. But yeah, they do lots and lots of sequencing. Now, one of the biggest things that we've seen, so flu sequencing we've done for a few, and that's been really interesting. They feed that into things like European networks, and that allows us to understand the sort of strains that are circulating, and that's been really important. Wales was up there right at the start of the COVID pandemic. Wales was up there as one of the top countries in the world for the amount of sequences that we had samples we had sequenced. So that's really fantastic. We had done a really, really good job, and that was thanks to the Pathogen Genomics Unit, which is sitting within microbiology. The same, we've got health protection sitting within the same organization, so you've got everybody together under one umbrella. And they were able to get some really, really good results out of that. But as a biologist, I'm, my bioinformatics isn't brilliant.
No, neither is mine.
Yeah. Yes, I leave that to somebody else.
Yeah.
It's not my strong point. I did try to dabble with it a little bit in my PhD, but yeah, not for me.
No, me neither. My brain just turns to mush with all of those like letters on the screen.
Yes, yeah.
So I know with other specialisms, what you learn at uni kind of reflects what you're doing in the workplace. So
Yeah.
is that the same for you? Because it just seems like there's so much that you cover in the workplace.
Yeah, so the master's program for microbiology, I know they differ across the disciplines. So microbiology is only offered by one university. It's in Queen Mary University in London, and it's 1 day a week, which is really, really challenging. I know a lot of disciplines are block release, but actually traveling up to London was, in the first 2 years, I would say the most significant challenge of the STP. It's a fair old journey. It's about 3.5 hours from where we are. Right at the start, we were trying to go up and back on the same day, which would mean that we were sort of getting up in the morning to leave for uni, getting back very late. We then moved to an overnight stay, but obviously that will then impact. Sometimes that would impact the rest of the time we'd had in the lab. Already taking a full day out for uni, you don't really want to lose too much more work time to the traveling. That was probably the biggest challenge with the university. In terms of the content, yes, we would have lectures on, right across all the different disciplines, but it would be, you'd have one lecture on malaria, and then you'd have one lecture on, respiratory infections or something like that. There is a lot of content to cover in it. Out of my colleagues that I work with, there's 4 of us. 3 of us have PhDs. So 2 of them were virology. Mine was bacteriology. And then the 4th one of us already had a master's and was from a bioinformatics background. So we'd all had sort of really different experiences coming into the program. And the clinical science program is very similar to a biomedical science program in terms of the content and the teaching. There are elements that are more clinically orientated, but it's still very much a laboratory discipline. Or there were certain things that I'd known from previous experience. There was lots of new clinical stuff that I'd learned from doing it. That was cut short during the second year at the start of COVID. So that's had a really big impact for us to have an opportunity really to put what we've learned into practice.
Yeah.
We came towards the sort of end of our second year, excuse me, and that's when sort of lockdown hit and COVID really started up, and that impacted on our ability to try and put some of those skills into practice, particularly the clinical side of things.
It sounds very difficult. I can't imagine having to go to uni every week for a day.
Yeah.
Like, that just doesn't seem like a sensible way of doing it on their part.
And what's even more frustrating is when you get people saying, "Oh, it'd be lovely. I'd love to go to London every day." A nice overnight stay, and you think, "Oh God, I can't think of anything worse."
Yeah.
That was a real challenge, and I was really lucky that I had a very supportive training officer. And I think that really is an understated challenge within the STP. People say about how challenging the STP is. Scientifically, I found it challenging, but I would say that the other elements have been the most challenging. Things like the amount of work in the amount of time. And I don't know what word I'd use, the emotional sort of impact of things like that, things like losing 2 evenings every week to work. The deadlines for the university, they would sometimes come bunched up, or they'd come at the same time as a rotation change, and there have been a few sort of pinch points where the pressure's really ramped up. I don't know if you found the same.
Yeah, I think it's not been too bad for us so far because we were able to do the first uni block from home. And so far, our deadlines have been a little bit more spread out, but I know for some of the second and third years they've got quite a lot on at the moment. And yeah, as you said, I don't think it's something I think the work, if you end up with like a good training officer and everything, they are understanding.
Yeah.
And they understand that you have got work to do, but I think uni can sometimes be a little bit more closed off to the other things that you
definitely.
have.
Yeah, definitely.
Yeah. What are your plans if you're coming up to the end of it now? What are your plans following the STP? Are you going to stay in a clinical scientist role, or are you going to look elsewhere? Because you've done a lot.
No, I definitely do want to actually work as a clinical scientist now. I think if I told my family that I was going to go and do anything else now, they'd lose the plot. My grandparents think I've been in uni for about 15 years. It's not quite that long, but yeah, I think it's time to finish school now and you know, to put a bit of this put a bit into practice. So yeah, looking to start in September as a clinical scientist, hopefully in Public Health Wales. Not quite sure what the roles would be yet. We've spoken about lots of different options. But I think that we're going into a really challenging period. I think the whole NHS is going into it. We're coming out of and going into a really challenging period. I think as we come away from COVID. Not to say it's going away altogether, but as we go away from that sort of pressure, there's going to be a new pressure of other diseases which have gone unchecked, maybe.
Yeah.
Or no, unchecked is the wrong word. We haven't given them the attention that they would normally have got, and I think there's going to be a lot of work to do there. I also think there's a bit of work to do around how we manage emerging tropical infections in future. It's brought a bit of a focus on microbiology and making sure that we've got those networks in place, and how we get those sort of early warning signs and do about it. Yeah, and I think that towards the end of my training now, the real focus is going to be on clinical contact. So people often wonder about the pathology disciplines, the blood sciences, the microbiology, the biochemistry, cellular pathology. I'll have missed some, apologies to pathology colleagues. About the amount of patient contact, and we don't have as much as some disciplines. I'm quite heavily involved with our trainee network, so I get to speak with cardiac sciences and the audiologists, and they have a lot of patient contact. Ours is a little bit more limited. We do still do things like ward rounds, so we go along registrars, looking to see antibiotic use and to check that antibiotics are being used sparingly but appropriately. Attending things like multidisciplinary team meetings. I've spoken a bit about that. And also sitting in on some specialist meetings, things like HIV resistance meetings, where you get to meet patients and things like that, which have been really interesting. But I think that's where I'm going to focus as I so the dissertation now is going to be in April, and then as I come into the final few months of my training, that's really the area that we're all going to focus on. We're up to scratch, ready for hopefully to get our registration and start practicing.
Yeah, it sounds so interesting because you see like all of the samples that are kind of written and read in massive letters at the top of our SOPs, like high risk samples and stuff, and you get them just all the time.
Yes. So most of the lab is a containment level 2 laboratory. In Public Health Wales, we do have a containment level 3, which is where you process some of the more high risk samples. Generally it was things like sputums, which can be more high risk, but there's lots and lots and lots, and I'm not going to embarrass myself by trying to list in them and running the list short, but there's lots of things which are handled at that sort of higher containment. Yeah. But you are right. Lots of things, pretty much everything that comes into microbiology will have high risk written across it. Not everything can be high risk, so then you need to work out what's actually high risk and what's just high risk.
Definitely. Well, that was such like a comprehensive view of microbiology. I feel like I thought it was this big and it's turned out to be like that.
Yeah.
So thank you.
It also smells a lot. You can normally smell your way to microbiology, whether it's the agar or the, you know, anything else. But yeah, not for the faint-hearted.
Definitely not. Brilliant. Well, good luck with your dissertation and the end of the programme. It'll be fine.
Thank you.
And yeah, good luck with what you carry on into.
Awesome. Thank you. Take care.
Thank you. Bye-bye.
Bye-bye.